Assalamu alaikum. Welcome back to the second lecture in this little mini series in our big lecture series, DNA and Design. And this little two-lecture series is about the topic of dysteleology and the issue of junk DNA. And in our first lecture, we basically defined junk DNA. We talked about dysteleology. We talked about how the issue of junk DNA was used as the smoking gun against intelligent design. And then we talked about how progress in science, beginning in about the year 2000, completely dismantled and overturned the notion of junk DNA, showing it to be a false notion. In this lecture, we want to now get into a little bit more detail. And there's really only so much detail we can get into for two reasons. Number one, that getting into much more detail would take us into the realm of genuine molecular biology, and that is an unbelievable amount of detail for the non-specialist. And number two, because we are at the cutting edge, there is so much that is still unknown, so much research now being done. Where research has now taken us is that most DNA is functional, and we're now just beginning to uncover some of the specific functions of the non-coding, what used to be called pejoratively junk DNA, and we'll explore a little bit of these topics in, again, detail that I would say for the educated non-specialist. So part of what is being discovered now is that the DNA, which does not code for protein, does indeed get transcribed into RNA, and that RNA is in charge of a lot of things, but one of the things it is in charge of is regulating which genes turn on, which genes turn off, and when and for how long those genes turn on. Now, this is important for two reasons, and we've already touched on one of them. Think, for example, about the different tissues in the human body. They all carry the same genetic information, yet they have very different functions. Your pancreas, for example, secretes insulin. The cells in your retina, on the other hand, are photosensitive and respond to light by sending out electrical signals. How is it that two cells that carry the same genetic information have such totally different functions? And this, of course, goes for muscle cells, blood cells, cells in your gut, cells in your liver, and so forth. Well, it is by regulating which genes get turned on and when. And an even bigger conundrum or bigger issue than the sort of static different functions of a heart cell and a brain cell and a liver cell and a muscle cell and a bone cell and a blood cell is how is it that these tissues came into being in the first place from an embryo that began as one single cell, and then it divides into two cells, and then they divide into four cells. All of them carry the same genetic information, and yet somewhere along the way, these cells then begin to differentiate into the various organ systems that work together in a coordinated way. For example, the endocrine system and the digestive system. Different systems, but when we ingest sugar, our pancreas secretes insulin. So somehow these tissues need to become differentiated from each other to form from a single cell at the beginning that carries one genetic blueprint, and not only to form as different tissues, but to form as different tissues that interact with each other in a coherent whole for a functional, healthy organism. And so for these things to happen, this complex regulatory network, this seems to be the function, or at least one of the functions, of the non-coding DNA, which gets turned into non-coding RNA. And now I just want us to get into just a little bit of detail about three different types of non-coding DNA. And let us begin by saying that non-coding DNA functions by making non-coding RNA. At least some of its function. We shouldn't make the broad assumption that that's the only thing it does, but at least some of the functions that we are beginning to understand is that DNA that doesn't code for protein, what was called junk DNA, does get transcribed into RNA, and then that RNA has the regulatory functions we talked about. And we painted it in very broad brush strokes. More detail than that, would require a very particular interest in molecular biology. But let's talk about three somewhat distinct but slightly overlapping types of the non-coding RNA. And so here's a paper in Genome Biology from 2017, and it is looking at the multidimensional mechanisms of long non-coding RNA. What is the function of long non-coding RNA? So the first class we want to look at are the segments of RNA transcribed from non-coding DNA that are long segment RNAs, and they are non-coding, and they're known in the parlance, if you will, among the specialists as link RNA, and link actually just stands for long non-coding, but you pronounce it link RNA. And so one type of non-coding RNA that seems to have very significant regulatory mechanisms, as the abstract of this paper says, is these link RNAs, or long non-coding RNAs, and they regulate gene expression through diverse mechanisms, as the abstract says. And for those of you interested in the detail, this is an open access paper. It's available to the public. You can just go ahead and pull the paper and enjoy the read. One recent result that came out of Harvard, for example, is that females have two X chromosomes, males have one X, one Y. Perhaps one thing that you are not aware of is that in females, one of the X chromosomes is inactivated. So even though females have two X chromosomes, they in general just express one of the X chromosomes. The other one actually gets inactivated and it moves to the periphery of the nucleus in the cell. And that is, for example, one of the functions of link or long non-coding RNA segments, and they are technically defined as RNAs that are 200 or more base pairs in length. So one type of regulatory RNA is long non-coding RNA, or link RNA. Now, another type of non-coding RNA that I want to talk to you about brings up a truly fascinating topic. And this RNA is the RNA which is found in introns. And remember, we had very briefly mentioned that DNA has regions which are expressed, and so they are called expressed regions. Expressed means they get turned eventually into a protein. And so exon is the abbreviation for expressed region. And then regions still in the same gene that are not expressed, and they are called introns. So the picture you may have had in your mind of a gene is a bunch of base pairs of DNA that all get coded into RNA, and then the RNA gets translated using those three-letter codons we talked about into a protein. Well, that was a first sort of brush painting, but the way that genes are actually organized are segments that are called exons, and you see in the diagram exon 1, exon 2, exon 3, exon 4, exon 5. In between them are DNA segments that are called introns. All of them get transcribed into RNA, and so that RNA will have a region that corresponds to exon 1, a region that corresponds to exon 2, and so forth, and regions in between that are intron RNAs. Now, what is the idea here? The idea here is really a magical and amazing idea that what this allows us to do, what this allows the human body to do, is something called alternative splicing. We said that the exons are the things that eventually go to make a protein. That means we have to take out these intron RNA regions because they cannot go ahead and code for amino acids, because then they would be expressed, and we just said they're not expressed. So the DNA gets transcribed to RNA, then the RNA regions that do not code for protein get spliced out. And so if you splice out the regions between exon 1 and exon 2 and exon 3, and so forth, then you would link all of the exons from 1 to 5 into a messenger RNA, and it is that messenger RNA that then uses the 3-letter codons to bring in amino acids, string them together to make a protein. Okay, wonderful. But it turns out that we have a mechanism within us called alternative splicing, so that instead of linking exon 1 to 2 to 3 to 4 to 5, I can, for example, drop exon 3. And I can then have a messenger RNA which is composed of exons 1, 2, 4, and 5. Now, when that gets coded into a protein, it's going to make a different protein called protein B in this diagram. And I can have yet another alternative splicing, one that would drop region 4, and I could use exons 1, 2, 3, and 5 to make yet a different protein. And I can even duplicate regions, not shown in this diagram, but I could have 1, 2, 2, 3, 4, 4, 5. And I could make yet a different protein. So you see the magic here, that there are now so many combinatorial possibilities that the same gene, by the method of alternative splicing, can express many different proteins. We always thought, and it was in fact one of the central dogmas of DNA that I was taught in college, that one gene equals one protein. We now know that there's this phenomena of alternative splicing, that I can take out introns and some of the exons and duplicate other exons and so forth, and so that exons 1 through 5 can make numerous proteins, not just one protein, depending on how we splice the RNA that gets transcribed out. And so this intron RNA between the exons seems to have a very significant regulatory function in deciding, working with other RNAs like link RNAs, which alternative splicing I am going to employ in my cell, and so which protein I'm going to make. And at one time, the cell, for a given need, can splice things one way and make protein A, and then when conditions change, it will change over in a dynamic process and make protein B or protein C or protein D and so forth. And so here's another function of these RNAs, now intron RNAs, an amazing phenomena. Okay, so we've now talked about two types of non-coding DNA and non-coding RNA, the link RNAs and the RNAs that are in introns that participate in this amazing phenomena of alternative splicing. And inshallah, when we talk about the information content of DNA in upcoming lectures, that's going to loom large, because we see now that DNA is not just one code, it is really a code within a code, because not only do you code for the amino acids that will make proteins, but you also code for when and how to do alternative splicing, and so how to use the same gene to express multiple different proteins. Now let's talk about yet a third type of non-coding DNA that was thought to be junk DNA, which is the so-called pseudogenes. Pseudogenes are a bit different than the other things that we've talked about because they look just like a known gene that codes for a protein, maybe sitting next to a known gene that codes for a protein, but they don't work, quote unquote, because they don't code for protein. Why? Because of a variety of things. They may have some mutation in them. They may have a segment that's deleted. They may be missing what's called a promoter sequence that allows that gene to be turned on and for transcription to begin and so forth. And so as opposed to the other types of RNA that we've already talked about, these pseudogenes are portions of DNA that look almost identical to a known functional gene, but don't end up coding for a protein. It now turns out, though, that these also do get transcribed into RNA and are a sort of pseudogene RNA. Now, why talk about pseudogenes? Well, because again, it was one of the big smoking guns for the evolutionary community. And I don't want you to get the idea that I'm anti-evolution. Inshallah, we will have a long lecture series dedicated to the topic of evolution. What I am against is random evolution without an intelligence behind it. But in any case, look at what leading biologists said about pseudogenes. They said that they are discarded sequences, quote, consistent with an evolutionary explanation, but inconsistent with intelligent design. That's from Kenneth Miller. We've encountered him before. And from Richard Dawkins, says that they are genes that once did something useful, but have now been sidelined and are never transcribed or translated. And Richard Dawkins, the very famous atheist who is also a famous biologist, says what pseudogenes are useful for is embarrassing creationists. It stretches even their creative ingenuity to make up a convincing reason why an intelligent designer should have created a pseudogene. Okay, so we've already talked about the long non-coding RNAs, the so-called link RNAs, and that their functions are now beginning to be discovered. And we've talked about the intron RNAs that are involved in this amazing phenomena of alternative splicing to allow one gene to encode for many different proteins. And pseudogenes are now going in that same direction. What were once thought to be just defective copies of genes are now being found to have very important functions. So here's a paper published in the journal Nature in 2008 that is saying that pseudogenes are transcribed and they give something called small interfering RNAs. And these small interfering RNAs are yet another type of functional RNA that regulates gene expression in oocytes in the very early process of embryogenesis. So in this paper, quote from the paper, that the findings indicate a function of pseudogenes in regulating gene expression by means of the RNA interference pathway. So this is a very particular way that we won't get into its details because they now are very complex in terms of the molecular biology, coding from the what's called antisense DNA strand and forming a double strand of RNA, which we thought never happened. We thought only DNA was double-stranded, and these double-stranded RNAs are known as small interfering RNAs, and they're able to interfere with genes selectively to regulate how they are turned on. And this seems to be important in the process of differentiation of tissues in the embryo. As time has gone on since that 2008 paper, more and more pseudogene functions are being discovered. This is just a blurb about examples of pseudogene function from the Wikipedia article on pseudogenes. Definitely feel free to look that up. But you see that, not only are there the short interfering RNAs, which are the first or the second paragraph rather in this blurb. Those are the siRNAs, but pseudogenes have turned out to give a lot of different types of RNAs, pi RNAs, microRNAs, and so on. And again, this area is just beginning to be explored, but what is clear is that pseudogenes are not just faulty discarded copies of genes that are kind of the litter and refuse and junk and remnants of the random evolutionary pathway, that they actually do have functions in gene regulation, and we're just beginning to find out those functions. So whichever type of RNA we look at, and again, a very important idea, we always used to think that DNA was turned into messenger RNA, and then it used so-called transfer RNAs and ribosomal RNAs to build proteins. Turns out that there are many, many, many different kinds of RNAs, most of them from non-coding portions of the genome, what was once called junk DNA, and we're now just beginning to understand some of the very complex regulatory functions that these RNAs have. And to end this lecture, I just wanted to share with you a couple of different quotes to make a couple of points. One is this quote from John Maddock, who, as we said, he is CEO of the British company Genomics, well-known molecular biologist. We've already seen at least one of his papers. And I just wanted to share with you his quote, that the failure to recognize the implications of the non-coding DNA, this idea that it's just junk DNA, will go down as the biggest mistake in the history of molecular biology. And I think that that's an important quote. And he really was instrumental in sort of recognizing that this human genome is not largely junk, but that there's the bottom quote, that this non-coding DNA specifies a massive hidden layer of regulatory RNAs that organize our development and provide the platform for brain function, among other things. And researchers at Harvard, the lab of Dr. John Rinn, for example, are beginning to now uncover disease processes that occur when there are mutations in the non-coding DNA, in these functional RNAs. Just like we looked at how sickle cell disease, for example, occurs with a mutation in one of the coding DNAs for the hemoglobin chain. Well, it's beginning to now be discovered that these non-coding RNAs are very important for human function, and therefore it stands to reason that when things go wrong with them, they will also produce disease. And this now is an area that is just beginning to be explored. What are the diseases, the genetic diseases, that are due to non-coding DNA errors that mess up the regulatory function of the various RNAs that we have talked about? So it's an amazing area, and we're living in a very exciting time, and really lucky to be people of faith who believed in intelligent design, because we have lived to see one of the main canons that the opponents of intelligent design were using against scientists of faith, the idea of junk DNA. We have seen that canon now get completely dismantled. In fact, turned around to not to go too much with this violent warlike metaphor, but to really be turned around and now can be used against those who are opponents of intelligent design. And I would like to end the lecture on one last point. And that point often goes by the wayside. The criticism leveled against those of us who believe in science and believe in intelligent design and believe, as the Quran has told us, that if we look in detail, we will see evidence of the intelligent design. The charge leveled is that intelligent design is unscientific. It goes beyond the boundaries of empirical science. Well, this is a beautiful case where we can see that intelligent design makes testable predictions. So the physicist David Snoke in 2001, really just as this notion of junk DNA was beginning to be dismantled, he has a long quote here. I'll let you read it for yourself. But the idea of the quote is that the entire idea of design, if we believe something is intelligently designed and you have a good design assumption, it will make specific predictions. And if you believe that something was assembled at random, that then also makes specific predictions. And as we saw, the notion of junk DNA was a specific prediction of a random cobbling together of some sort of DNA that would work to allow life, but it shouldn't really be sophisticated and well designed because random processes don't in general do that. And then the quote by the chemist Jonathan Sarfati was saying that junk DNA, this idea, quote unquote, junk DNA, is not evidence for evolution. Rather, its alleged junkiness is a deduction from the false assumption of evolution. Just because no function is known, it doesn't mean there's no function. And so we see long before the junk DNA hypothesis was dismantled, scientists who believed in intelligent design predicted that even though it seems that this DNA is non-functional, if we believe in intelligent design, we have to believe that it will turn out to have a function. So Michael Denton, a physician and biochemist, if you look at his quote in 2002, says, if it were true that the genomes of higher organisms contained vast quantities of junk, then the whole argument of this book, i.e. intelligent design, would collapse. Teleology would be entirely discredited. Teleology, again, links to that term, dysteleology. It is the underlying purpose, the underlying deliberateness behind why something is created. And he is saying that on any teleological model of evolution, most or perhaps all the DNA in a genome of higher organisms should have some function. So this was a testable scientific prediction that the theory of intelligent design not only can make, but did make years before the junk DNA hypothesis was discredited. And let's look at biochemist Michael Behe, who has done tremendous work in the science of intelligent design. He was responding to Kenneth Miller's arguments for junk DNA that we've already seen. And he was saying that because we have not yet discovered a use for a structure does not mean that no use exists. And William Dembski, another huge figure in the intelligent design community, mathematician and philosopher, is said outright, on an evolutionary view, we expect a lot of useless DNA. If, on the other hand, organisms are designed, we expect DNA as much as possible to exhibit function. And this was in 1998. This was at a time when a man of faith like Francis Collins, who was head of the Human Genome Project, was still subscribing to the idea of junk DNA and that it supported the Darwinian evolutionary mechanism. So proponents of intelligent design were making a very clear statement that here's a testable hypothesis. If it turns out that indeed this DNA is junk, then that would support the Darwinian evolutionary mechanism. If it turns out that it has function, that would be much stronger support of the intelligent design hypothesis. And this is exactly what we have seen happen in the last 20 years. So I hope, inshallah, that you are finding these lectures interesting and instructive, and that um, you are hopefully convinced with me that there is value to digging a little bit into the detail. It is one thing, and it is not a small thing, to look at the beautiful blue sky and say, Subhanallah, this is so beautiful. It is another thing, another level of depth, to understand the quantum mechanical reasons as to why the sky is blue. Both of them are valuable. Both of them are important. Both of them are a gateway to faith. And I hope that you will keep joining me for the rest of this lecture series, where we will tackle other important issues about DNA and design. Take care. Assalamu alaikum and God bless.